Alrighty, so this is for STAAR review number 2, cells part 1. First thing about them is the types of the cells. So if you look at the picture that you have right here, you've got 3 different cells in the picture. The first thing you notice is that these 2 cells right here are way more complex, and this one is a little bit more simplistic. What does that mean? These 2 right here are eukaryotic cells. This one right here is prokaryotic. Now remember, prokaryotic, eukaryotic, what are the big differences? Eukaryotic cells have a nucleus and they also have membrane-bound organelles. But there's 4 things that all cells have in common, technically 5 things. The first one is they all have DNA, they all have the ability to maintain homeostasis, they all have cell membranes, cytoplasm, and oops, forgot the last bullet point on there, ribosomes. How are you going to remember these 5 things that all cells have in common? You're going to say, do not hate runny chocolate chips. Alrighty, so do not hate runny chocolate chips. That'll help you remember. You might also see it like this, just DRCC. A lot of people don't include the homeostasis. So again, do not stands for DNA. Hate stands for homeostasis. The R stands for ribosome. C stands for cytoplasm, and then cell membrane. So do not hate runny chocolate chips. Alrighty, the next section of your notes, well, it's actually in the same part, is this picture. And what is this picture standing for? It's standing for the theory of endosymbiosis. Remember, the word endo always means within. Symbiosis means a relationship. So what is this talking about? It's saying, how did we form the very first eukaryotic cell? What happened was a small prokaryotic was swallowed by a large prokaryotic, and instead of the large one killing the small one, it actually lived inside of it, and they formed some type of relationship. Because they formed a relationship, that became the very first organelle. Therefore, this became the very first eukaryotic cell. Alrighty, the next section, so this is going to be eukaryotic cell organelles. Because I've got a plant cell and I have an animal cell. So these are our 2 eukaryotic cells, and we're looking at the organelles inside of them. Remember, an organelle is a small little package that carries out a function. So the ones I want to point out to you, obviously we've got the nucleus. Remember, the nucleus contains DNA. That's also where the first part of protein synthesis will happen. I do want to point out the chloroplast, which is only going to be located in plants. Remember, the chloroplast carries out the process of photosynthesis, which we'll talk about in the next video. The ribosomes, obviously they produce protein. The next one is the endoplasmic reticulum. There's 2 parts to the endoplasmic reticulum. There's smooth and rough. If it is the rough endoplasmic reticulum, it's covered in ribosomes. So what is its job? It helps with protein synthesis. Smooth just helps with other production of molecules. So those biomolecules we learned about, the smooth endoplasmic reticulum helps with that. Once those molecules are made in the smooth and rough, they actually travel to the Golgi apparatus, and then from the Golgi apparatus, they're transported out. So they might be transported to another cell or to another part of that cell itself. Obviously, the cell membrane, its job is to keep everything contained and in one location. It also controls things that move in and out. And then when we talk about the vacuole's job in plants is to store water, nutrients, and waste. It's very large. So we say plants have a large central vacuole. Animal cells will also have a vacuole. It's just going to be a little bit smaller because we don't really need to store that much water. Of course, we have our cytoplasm. Cytoplasm's job is to actually make sure that it's the gel, to make sure that everything stays in one location. Plants and some types of animal cells, so for example fungus, will have a cell wall. That cell wall's job is mainly to just give it structure and support. And the last one I want to talk about is going to be the mitochondria. Mitochondria, remember, is the powerhouse of the cell. It goes through cellular respiration. So before you leave this page, make sure you have the definition of what these organelles do. You can pause the video, rewind it, whatever you need to do. Mitochondria carries out cellular respiration. Chloroplast carries out photosynthesis. Those are 2 big ones we're going to talk about in our next video. Alrighty, your next section is cellular transport. Cellular transport, basically the purpose of this is to move stuff in and out to maintain balance. Remember the word balance, homeostasis. And remember, all cells, prokaryotic and eukaryotic, move things in and out. So this is happening in both. The first type of transport we're going to look at is passive. Passive, another name for that is diffusion. We're moving things from high to low. So in this picture right here, it's a high concentration moving to a low concentration, passive. This picture right here, high to low, but it's got a protein channel. So this is called facilitated diffusion. It's still same passive, doesn't require ATP or anything like that. It's just using a protein. Our next last picture is going to be for active transport. Active moves from low to high. Notice I'm using ATP. Why? If I'm moving something from low to high, I'm basically squishing it onto a side where there's already a lot of it. So that requires energy to push it in there. So ATP, active, they both start with the same letter, easy to remember. Let's go ahead and look at this graphic. This will help you. I suggest you write this on the corner of your test as soon as you get it. Down here is low concentration. Up here is high concentration. So these are like the low parts of the mountain, and this is like the high part of the mountain. If I'm down at the bottom of a mountain and I have to bicycle, I have to pedal up a mountain, obviously I need to use ATP. So if I'm pedaling up a hill, I'm using ATP. What type of transport? That would be active. If I'm at the top of a hill and I want to go down, I don't use any ATP, so this will be passive. So just as a refresher, going from high to low, passive. Going from low to high, active, and that requires ATP. Suggest you draw this. Definitely label it on your picture. Make sure you draw that on your test. It'll just be a lot easier for you. Alrighty, we're going to look at one example of passive transport, but this is the movement of water. So again, it's passive, so I'm moving water without using energy. What is this called? This is going to be called osmosis. For osmosis, I'm moving from where there's low salt or sugar to where there's high salt or sugar. So think about it this way. Every time you eat something really salty, you want to drink something. So water always follows the salt or the sugar. So let's look at an example. If the outside of my cell has low salt or sugar, the inside has high salt or sugar, water will move to where there's high salt. So it will move in. If too much water moves in, my cell will eventually get so large that it will burst or break. We call that lyse. So the word lyse means burst or break. The next type of solution water moves in would be isotonic. Iso means equal. So that means that inside, for example, there might be 5, outside there's 5, and so water moves in, water moves out at an equal rate. This is normal. This is what we want. This is ideal. The last type of solution is called hypertonic. Hyper means above. So that means the outside has high salt or sugar, the inside has low salt or sugar. Again, water follows the salt, water leaves your cell and it shrivels up. This is what happens when you are exposed to the Dead Sea. In the Dead Sea, it's high salt on the outside. Your water will literally leave your cells and you'll basically just shrivel up and die. By the way, going back to hypotonic, hypo means below. Hypo, below, hypo, below. So keep that in mind. Hypo means below, hyper means above. This is an example of a plant cell and what it looks like. So for hypotonic, where the water's moving in, your cell gets very large and weird looking. It's usually called turgid, and that's a good thing. We want our cell to be turgid so that way it stands up straight. In isotonic, where water is moving equally in and out, we call that flaccid, and that's okay. It's not good. It's not great. We want it to be turgid, though. And our last one for plants is where it's hypertonic, so high on the outside, low on the inside. All the water leaves. Our cell will basically shrivel, and this is what happens when flowers die when you don't water them or something. They shrivel up. The next one is an example of active transport. Remember, active uses ATP. I'm going from low to high, and I'm using ATP. There are two types, endocytosis, exocytosis. Endo means enter, so endo, enter. Exo, like exit. So basically for both of these, I'm using a vesicle, which is cell membrane, to either move stuff out, so that's exo, or to move stuff in, which is endocytosis. Why do I use ATP to do this? Because I'm moving a lot of things all at once. I'm not just moving one molecule at a time. I'm moving like 60, 70, 80, 100 molecules all at once, or I'm moving really big, large molecules. Think about it this way. If you had to carry 60 textbooks all at once in, it's going to be very heavy for you. So endoexocytosis, moving a lot of things using energy. Our very last section is going to be for protein synthesis. Remember, the word synthesis means to build or to create. So what am I building? I'm building protein. This is a two-step process. If we look into eukaryotic cells, remember we have a nucleus. So the first step, which is called transcription, happens in the nucleus. So eukaryotic transcription happens in nucleus. For prokaryotic, they don't have a nucleus, so it happens in the cytoplasm. What is transcription? Transcription is the first part of this, where I'm reading DNA and I'm building mRNA. Who does this? So let's go ahead. This is this process right here and this process. Let's go ahead and look at this diagram to go ahead and see how it happens. So remember, a double helix, that's my DNA. Single strand, that's my RNA. Just a little reminder, DNA and RNA, how are they different? RNA has U instead of T. Okay, so it doesn't have T, it has U instead. So notice how every time there's an A, instead of matching it with T, I match it with U. So RNA polymerase is the name of the enzyme that's going to be reading DNA, building mRNA. What direction does it build in? It builds 5 to 3. Remember, DNA is directional. Okay, so there's a 5' end and a 3' end of DNA. Well, RNA polymerase always builds 5 to 3. So we start by reading my DNA, building 5', 3'. This process always happens wherever the DNA is located. Just keep that in mind. Our second part is called translation. It's going to happen wherever the ribosomes are. Well, in both prokaryotic and eukaryotic, ribosomes are always going to be located in the cytoplasm. So this happens in cytoplasm for both. Ribosomes, remember, they build the protein. What do? They actually read mRNA and they build the protein. Remember that chart we use? That chart basically has all the letters that we're going to be using for the names of the amino acids, and we'll practice that in class.